All right. Hello, everyone. Well, first of all, I would like to thank the Edelweiss Connect team for organizing this webinar series. This webinar will be about the SafeDESIgn image-based reporter platform that we can apply for the chemical safety assessment. I'm Braak, and I'm currently working at the Leiden University. But if all goes well, we hope that we could start a spin-off company based on the technology that I'm going to talk about today. And then hopefully I can work there. So I think this webinar will be around 30 minutes, so there will be plenty of time to questions, discussion, and answers. I would like to start with a brief introduction in the toxicology field, because our body consists of cells, and these cells would try to remain in homeostatic phase. So basically, this means a stable external and internal environment. And obviously, this is disrupted when there's a chemical exposure. So the chemical exposure will lead to harm to the cells, and the cell will try to counteract this harm. And therefore, it will activate cellular stress pathways. To give you an example, is the oxidative stress pathway, in which free radicals are actually causing the harm. The cell will try to counteract this by activating the NF2 signaling cascade. And then antioxidants will be produced, and these antioxidants then counteract the cellular stress, restoring homeostasis. Now, this goes well for low concentrations, but at higher concentrations, the cell can't cope anymore. There's simply too much injury, and instead of investing into these adaptive stress responses, the cell will try to invest more into adversity programs like necrosis, autophagy, and apoptosis. And this will eventually lead to dead cells, a damaged organ, or a sick person. Now, in Leiden, we would like to understand this whole process. And of course, there are different so I already introduced the oxidative stress pathway, but obviously there are more of these adverse and adversity stress signaling pathways, like DNA damage, ER stress, also called the unfolded protein response, inflammation, heavy metal stress, hypoxia are just a couple of them, but there are way more. And if you want to study these responses, there are different ways. So you can, for example, make use of Western blotting, RT-qPCR, stainings, but these are very variable, so a very large variation, and they're really labor intensive, and it will just cost you a lot of time. So in Leiden, we think that a different way of studying these very dynamic responses, and we have chosen the reporter model. So about the generation of this reporter model, we started with the HepG2 liver cell line, and then we transfected with a very big plasmid on which our biomarker, so we select a certain biomarker for a certain stress pathway, and this is located in this bacterial artificial chromosome, or short BAC. So basically at the C terminus, so right before the stop codon, we clone in a GFP sequence. So whenever the biomarker is being expressed, also the GFP will be expressed, and then a GFP fusion protein will be formed. And this is very, very important, because whenever, so in this case, we can not only see how much of the biomarker molecule is available, but also where in the cell it's located. That's very important if you want to make, for example, conclusions on activation of a certain molecule. So, for example, transcription factor is only active in the nucleus. So if you quantify the amount of nuclear transcription factor, you're looking at active transcription factor. All right, so in total, we have done it for a couple of these stress pathways. And on the left, you see, for example, the oxidative stress pathway. And every time we try to tag an early regulator, in this case, KEAP1, the transcription factor of the pathway, NF2, and also its downstream targets, so sulfur-redoxin, NQ1, and HEMOX. And these are all examples of these earlier mentioned antioxidants. So in total, we've made quite a lot of these stable reporter cell lines. In total, we have around 60 of them, and these cover the majority of these stress signaling pathways. All right, so how does the screening setup looks like? So we start with seeding our reporter cells in a 384 well format, as you can see over here. So it's quite high throughput. Once the cells have attached, we add a host, which is a nuclear counterstain. And then, so we can also do single cell tracking with the host. And then we add our compound at a certain concentration. And then every hour we take an image. Well, if you take every hour an image, you can make a time lapse. And you can really follow what is happening over time with your stress response. So if we right now look at the left panel, you can see some examples of the DNA damage response. And over here, P53-GFP, the stable report cell lines presented. And if we add DNA damage, you can see, so for example, with cisplatin, you can see that there's accumulation of nuclear P53 over time. When the P53 is in the nucleus, it will actually activate downstream targets, and BTD2 is one of them. Note that the subcellular localization of BTD2 and P3 is very different. So P3 is only in the nucleus, whereas the BTD2 is also in the cytoplasm, as you can see. On the right side, we see a different reporters over different pathways. So these are the inflammation reporters. So REL-A is a subunit of the NF-κB signaling response, a well-known inflammation pathway. And if we induce this pathway with, for example, TNF, you can see that normally the REL-A or NF-κB is in the cytoplasm, but directly after we add TNF, so in the first half an hour, we see that the TNF, the REL-A translocates to the nucleus, where it acts as a transcription factor. And then it will activate downstream targets like ICAM, but also A20. And A20 is a negative regulator of the REL-A, and this creates this oscillatory pattern of the REL-A or NF-κB translocating to the nucleus, but then also it's being dragged out of the nucleus into the cytoplasm and into the nucleus again. So you can create this oscillatory pattern. And I think there's a beautiful example of how we can actually study such a very dynamic response with our reporter setup. I don't think there's any other readout in which we can actually visualize and quantify such a dynamic process with this high quality. All right, so from a simple screen, we generate about 30 terabytes of data. And of course, we cannot just quantify all these responses by hand. So luckily, people in our lab have written some image segmentation software to do this automatically. And basically how it works is that we use our nuclear counterstain, the host, to really identify where all the individual cells are. From that, we also propagate a cytoplasm, and then we make an overlap with the GFP channel and the segmented nuclei and cytoplasm. And then we can do really, on a single cell level, we can quantify the amount of GFP that's in the nucleus and the amount of GFP that's, for example, in cytoplasm. For some of the reporters, the localization is not per se the nucleus or the cytoplasm, but they form these speckles. So later on, I will give you an example of this, but then we can also quantify the number of speckles per cell and also the intensity of these speckles. And from that quantified data, we can make nice plots, so time response plots, concentration response plots, and do some feature extraction. So for example, we can generate points of departures. This is actually the first concentration at which we see a significant induction of a certain pathway. We can actually extract EC50 values. The slope can be determined, or maximum maximum intensity levels can be extracted. And from some of the reporters, as already mentioned, single cell tracking is really necessary to have these single cell features. For the REL-A, the NF-κB, reporter. This is an example. Then we can really estimate this oscillatory pattern as mentioned before. All right, then we'd like to highlight one of the stress pathways, because later on in the presentation I will show you some applications of the BAC-reporter system, and then some basic knowledge on this pathway is necessary. So over here we see a normal cell, and there's a normal cell in unstressed conditions, so homeostatic conditions. And in homeostatic conditions, we see that the KEAP1 is bound to the NF2. And when this happens, the NF2 is polyubiquitinated, and it will be targeted for proteasomal degradation. And this just to make sure that the NF2 pathway is not being turned on. But when we add oxidative stress, for example with drug, then the sulfide bridges at KEAP1 will be hydrolyzed, and then KEAP1 will be polyubiquitinated itself, and it will be translocated in these small foci, which are actually autophagosomes. And that's what you see over here. So in the KEAP1 reporter, the time lapse, you can really see that all these small speckles. So these small speckles are really KEAP1 foci accumulating in these autophagosomes. So when this happens, NF2 is being released and will accumulate in the nucleus, and that's what you see over here. So NF2 is a very weak reporter because NF2 is hardly expressed in the cell as most transcription factors, but you can see that the response is still quite good visible, and whenever we add oxidative stress, you can see clear translocation accumulation in the nucleus. When this happens, of course, they will bind to antioxidant response elements, and this will ensure expression of downstream targets, HEMOX, NQ1, and sulfadoxin, the antioxidants that I was talking about earlier. And you can see very clear induction of biomarker GFP levels in this case. And then we can, of course, quantify this and create these dynamic plots. A question that I get quite a lot is how important is the dynamic factor? So if you would have just picked one time point, would this make a big difference? Well, sometimes not, but what we see sometimes also is that, and this is again the NF2 pathway, and we determine nuclear NF2 in this case, and we exposed with two compounds, and I think these are the two most well-known inducers of oxidative stress, sulforaphane and CDO. And what we see is that the time dynamics of the NF2 response is very different between these different compounds. So for sulforaphane, we see that there's a very clear induction and a peak at 6 hours, but then also steep reduction of nuclear NF2. And this is very different from the CDO response. So there's a somewhat weaker response, you might say, but it also is gradually inducing over time. And this is probably due to stability of different stability of the two different compounds. But there's a big difference. And what we actually find is that if you take the area under the curve of these plots, you'll get quite a good estimate of how much oxidative stress a certain cell is experiencing. All right, and I would like to show you an example of how we use these BAC-reporters in fundamental research. And for this, I already introduced the NF2 pathway. So again, KEAP1, NF2, and we can actually add reactive oxygen species that interact with KEAP1 cysteine molecules. And then more KEAP1 will be degraded, NF2 will be released, and the pathway will be activated. So this is what we already know from literature. But what we do not really understand is how this is regulated. So we know that on a post-transcriptionally level, this is being regulated by, for example, ERK and JNK. On the translation level, microRNAs might be involved. Transcription level, there might be factors that might affect the NF2 pathway, but also as binding partners, they can directly bind to NF2 and then affect this pathway. But what these factors actually are is not really known. So therefore, we used our BAC-reporter system, and at first we focused on the sulfadoxin cell line BAC-reporter. And in this BAC-reporter, we knocked down with small interfering RNAs over 3,000 different genes. And then we activated the NF2 pathway with low concentration of CDO. And we determined the whole dynamics of the sulfatoxin to really see if the knockdown of the certain gene had affected the NF2 pathway. And actually, to our surprise, we found that over 180 different genes significantly affected the sulfatoxin responses. And of course, there were some logical genes. For example, key1 is a logical hit because if you knock down the negative regulator of the NF2 pathway, you expect the sulfatoxin levels to go up. So that's nice that we also see this. NF2 on the other side, if you knock down NF2 itself, you do not expect very high levels of sulfatoxin. That's again what we see over here. But besides these two genes, there are 180 more genes that significantly affect this pathway. And right now we are involved in, so then we also performed a deconvolution screen. So first we screened the SMART pool, so combination of 4 small interfering RNA sequences targeting the same gene. And right now we also performed deconvolution screens, so screened also the single RNAs to really validate some of these genes. In total, we validated in this case dozens of different genes. And right now, these are being screened with other assays as well. So pull-down assays, for example, to really see if there's a physical interaction between, for example, NF2 and these targets. So that's still going on, and that's how we apply the BAC-reporter technology in fundamental research. And yeah, by doing so, solving more or less the puzzle of the NF2 pathway. All right, then the second example of how we apply these reporters, and that's in modeling. Because there's quite some interest also from the industry in these modeling approaches, because they think it's a nice in silico tool to actually predict with just some very, very small chemical descriptors what the biological effect will be in a in vitro setting, or hopefully even an in vivo setting. So for this, we focused on the p53 pathway, and we included 5 different reporters. So 53 binding protein 1 is an early regulator of this pathway. p53 is the transcription factor, and MDM2, BAC-GFP, PTEN1 are 3 downstream targets. In unstressed conditions, there's no activation of either of these biomarkers, but if we add oxidative DNA damage, in this case is done with cisplatin, you can see that the pathway is being activated. So we can see speckle formation of 50 binding protein 1 in the nucleus, and it's pretty cool because 50 binding protein 1 really binds to these double-stranded breaks. So that's really what you see over here. Nuclear p53, nuclear MDM2 and PTEN1, and BAC-GFP is, as mentioned earlier, in the cytoplasm as well. And for this, we screened 72-hour live, and we also included some cell death markers. And Muriel, one of our PhD students, she is really looking into this, and right now she's forming these ordinary differential equation models, ODE modeling, to really predict how much of these biomarker proteins are in proliferating cells, how much are in arresting cells. And so right now she's still focusing on the first 4 concentrations. So these are below the cytotox concentrations. And right now she's checking, is her model able to predict these biomarkers? And for the first concentration, it works pretty well. So here's the experimental data, in which you see very, very nice dose-dependent induction of these pathways, these biomarkers. And this is the qualitative fit, so her ODE model predictions. You can see it nicely fits with the actual data. And this is just with a very simple ODE model, so it's pretty quite amazing. So the next step is also to include concentrations at which we actually do induce cell death, and hopefully eventually we can have this model in which with some very simple characteristics of the compounds, we can actually predict what is going to happen in an in vivo situation. That's the end goal. All right, then the last application of the BAC-reporters, and it's in hepatotoxicology, so basically liver toxicology. And for this, I will show you this small screen. The small screen consists of 14 different compounds, 4 negative control compounds and 10 compounds, in which we know that in vivo, so in the human situation, liver tox is being induced. So there are many companies that claim that they have very sensitive models that can study DILI, drug-induced liver injury. And the majority of these are actually focusing on one endpoint. It's a classical endpoint, cytotoxicity. So basically what they do, they have their cell model, they expose to a certain compound, and they just measure the fraction of cell death that is being present. And obviously, this will tell you something about how toxic this chemical at a certain concentration is in your cell model. But it's not per se giving a lot of information about what will happen eventually in an in vivo situation. And that's nicely depicted over here. So if we just focus on cell death readout in our assay, you can see that only 4 compounds are being classified as being liver toxic, and we miss 6. So if you just focus on the classical endpoint, you miss 6, so 6 false negatives. But obviously we have way more data. So if we include also data of the sulfurredoxin as a measure of oxidative stress, the CHOP GFP as a measure of UPR response, the HepG2 PTN1 BAC-reporter data as a measure of DNA damage, and this is the ICAM1 reporter as a measure of inflammation. You can see just by eye, just by looking at these plots, you can see that we correctly could classify the compounds that would induce liver tox and the ones that do not. Well, that's nice that we can predict it for 14 compounds, but can we also now predict it for a larger compound screen? And for this, we collaborated with the FDA, with Weira, Tong, and Mignon, because they wrote 10 years ago already a nice article in which they actually mentioned 170 compounds, and they labeled all of these. And the labeling went from severe drug-induced liver injury compounds to no drug-induced liver injury compound drugs. And of course, the question is, can our reporter model correctly classify this labeling, basically? And for this, we included first 4 different reporters, BAC-reporters. So the HepG2 ICAM GFP reporter, oxidative stress, PTN1 DNA damage, CHOP, unfolded protein response, and sulfurredoxin as a measure for oxidative stress. And what we can already see from this hierarchical clustering, I'm not going to really dive into detail over here, but over here you see all the different compounds, but there is an enrichment of most severe DILI compounds over here. And this is also where the most of the stress pathway activation is happening. So just by looking at the data, it looks that there's at least an enrichment. So the second phase, we actually included more reporters. So besides sulfurredoxin, we also took a second oxidative stress reporter, HEMOX. We also included PTN1 next to PTN1. We also included BIP next to CHOP as an indication of the unfolded protein response. And we also included this heat shock protein. And then with linear discriminants, we tried to predict basically how well we could predict the DILI labeling. And in fact, in 66% of the cases, we were correct, which might not sound like a lot, but it's already way better than the current models that are focusing on the classical endpoint cytotoxicity. But obviously, we're not satisfied yet. So we thought, okay, maybe the relation between these activation of networks and DILI is more complex and is not linear. So we also tried other approaches, and over here the neural network machine learning approach is being presented. So what we did is we split our data set into 2. So 70% we actually used to train our neural networks, and the remaining 30 was then used to validate our prediction. And this was iterated then 100 times, and eventually we ended up with a total prediction score of 70%, so slightly higher than the method with linear discriminants. So 70% is already quite good, but we obviously have way more data. So besides the stress pathway activation, we can also include data on cytotoxicity. So we have PI, and which is a marker for late apoptosis and necrosis, and then XF5 is a marker for apoptosis, which can be simultaneously added to actually the screening setup. But besides that, we also have information on the compounds. So, for example, the molecular weight, the log P, electronegativity, or polar surface area, all kinds of chemical descriptors that we already know from these chemicals. And by combining all this data, by combining this also with our neural network approach, we eventually could get a very nice prediction. So a sensitivity of 96%, a specificity of 82%, and a total accuracy of 88%, which is of course a very, very big improvement if you compare it to other assays that claim that have very good predictivity on DILI. All right, so the data that I've just shown was all based on the 2D methods. So basically culturing these HepG2 BAC-reporters in 2D plastic surface. But we can also culture these in a 3D matrigel environment. So basically what we do is we see the cells in a single cell manner, and during around 3 weeks, these stable spheroids are being developed. And these spheroids consist about 1,000 individual cells, as you can see over here. And what we can also see is proliferation stops. So from day 21, these spheroids are quite stable, and it's quite nice because then we can also do repeated dosing, which of course quite interesting for hepatotoxic screens. Another nice thing is that hepatocyte-like features are being induced during differentiation, during the formation of these spheroids. So 2 examples. So, for example, the P450 enzymes are being induced, and that's what you can see over here. So in 2D, there's hardly any CYP2E1 expression present, but during differentiation, you can see that there's a strong induction of CYP2E1 expression. And by the way, these P450 enzymes are really important for in hepatocytes, because hepatocytes normally convert drugs and detoxify drugs. But sometimes we also see is that some compounds, the mono compound is not toxic, but the metabolite is toxic. So if you miss the enzyme that's converting the mono compound in a toxic metabolite, you might have a false negative. So therefore, this CYP expression is quite important. Well, besides P450 enzyme expression, it's also nice we have expression of these transporters. So UGT transporters, the family name, but there are way more than just this one. But you can see for this specific transporter, see very nice induction when the spheroids are being formed. All right, so the last couple of years we've put quite some effort in making this system more robust. So we use, for example, robotics to actually add the matrigel, then add the cells, and then after 21 days we have mature spheroids that can be exposed to the drugs. And for this we use also a liquid handler, so robotics to actually add drugs. And by doing so, we were actually able to really reduce variation, and that's what you can see over here. So we have the 384 well plate, and you can see all these small spheroids over here, and there is some variation in spheroid size, as you can see over here, but it's quite reasonable. And also with segmentation, we can, for example, discard this smaller spheroid. All right, so the big question is, of course, how does the 3D system perform? And at first we focused on the issue, is repeated dosing making a difference? And for this we screened 2 compounds that are actually falsely negative in our 2D screen. And in a 3D system, you can see that there is actually a dose-dependent induction of this specific compound, and For this, we actually focused on sulfur-redoxin GFP, so oxidative stress. And also for the second compound, trazodone, we see that at the highest concentration, there's a significant induction of sulfur-redoxin GFP. But if we right now do a repeated dosing, so instead of a single exposure of 48 hours, we dose every day and then for 6 days. And all of a sudden, we see a very, very clear dose dependency, which is actually kind of missing in the single exposure. So yes, repeated dosing does make a big difference. All right, so that was for 2 compounds. So right now we expand the panel a little bit. So for some of the compounds, we did not really induce cell death in 2D. So for example, for aspirin, we never see actually really a strong dose-dependent induction of cell death. And the same is true for the 3D situation. So in this case, the 3D situation did not make a whole lot of difference. But for example, for trovafloxacin, as one example, we see that if you culture in 2D, the cells are not responding at all to the trovafloxacin, at least not when it comes to cell viability. But if we culture them in 3D, we see that there's a dose-dependent reduction in cell viability. But if we now add a repeated dosing, we see that the system becomes very, very sensitive. And it's quite logical because trovafloxacin itself is not really toxic, but CYP3A4, so one of the CYP enzymes, metabolizes trovafloxacin in a toxic metabolite. And this is quite not a very stable metabolite. Therefore, the difference between 3D and repeated dosing is very big. So this is what we see for trovafloxacin. Also for some of the other compounds, we see that the 3D repeated dosing is more sensitive. All right, so for the last few years, we have been collaborating quite a lot with industry. Cosmetic industry, chemical industry, food industry, and I can only disclose 2 names. So we've worked together with Syngenta and in Unilever because these 2 projects are also partly published already. For other industry partners, we have not yet or will not publish the data. But what I can mention already is that for some of these projects, we are already in the second or third project. So apparently, the industry is quite happy with the results they receive from us. And we think also right now that the technology is ready to actually be spun off from the university, so we can start a spin-off. And we already have a name. DESIgn, short for Drug Efficacy and Safety Imaging. And for the last few years, we've also visited some conferences and congresses to really get some exposure, to really see what the demands are from industry. We talked with industry. And this also led to some of these collaboration that right now we perform under the flag of the university. So if you're interested in DESIgn and what we might offer in the future, I encourage you to visit this YouTube movie in which we actually describe what we do and what our vision is. All right, so how can our technology called the SafeDESIgn platform help the industry? So on the left, you can see the well-known drug development funnel in which 10,000 compounds are being put in, and eventually, after 15 years and spending €1 billion, eventually, hopefully, one medicine will reach the market. Very inefficient system. So hopefully, SafeDESIgn platform can help with this and make it shorter and make it cheaper, and hopefully, especially in the first part of the preclinical in vitro screening, I think the SafeDESIgn platform can really make a difference. And with different products, so for example, the SafeDESIgn Tox2D is a product in which we can screen at one time point. It's quite high throughput, so we can handle thousands of compounds in one screen. Then we also have the SafeDESIgn Dynamics, in which we really can follow over time what is happening with stress pathway activation. And we can screen for 24 hours, but if it's really necessary, we can also screen for 72 hours in the 2D system. And well, obviously, the plate, the 384 well plate, will be under a microscope for this period of time. So this will also lower the throughput a little bit, but still we can handle hundreds of compounds in this case. And for the last phase of the preclinical in vitro step, so it will be step 3, I think that the SafeDESIgn Tox3D will be very suitable, in which we create these nice reporter spheroids, and we can actually do repeated dosing and image after, for example, 6 days of repeated dosing. All right, so I hope this is all clear, and if not, then I would like to get some questions from you. You can either email me or reach out via Edelweiss Connect. But first I would like to thank the people that have been involved. So people from Leiden University, Bob de Water, Professor Steven Wink, Steven Hiemstra, Marije Niemeyer for co-developing the BAC-reporter model. Wouter den Hollander, Lucas Muriel Olivier, and Joost Belman for actually the modeling part that I showed in my presentation. These I'm currently running that together with Bob van de Water and Lisanne Wolters, a very good technician. And Weide Tong and Minion from the FDA for actually providing us with the big compound lists from the FDA with a clear drug labeling. So yeah, thank you. And if there are any questions, I will be happy to take them.